Okay, so today we're going to cover our topic of Unit 1, which was all about cells. So let's dial it way back to 2 years ago when we started off talking about cell theory, right? And so y'all probably know it by heart by now. That cell theory is a theory that all cells are made of at least one or more cells, that the cell is the smallest unit of life, and that all cells come from pre-existing cells. There were some limitations to cell theory, right? So these were the four quote unquote exceptions. So they included things like striated muscle and aseptate fungi, which are kind of like fungus roots. And so for both of them, there are limitations to cell theory because they have many nuclei, they're multinucleated. And it also included giant algae, which was really large compared to a normal cell. And then there was also red blood cells, which have no nucleus and therefore cannot replicate on their own. So those were the limitations to cell theory. We also know that cells, apart from the giant algae, cannot get too large, and that is because as the cell volume increases, surface area does not increase nearly as fast. And so what we're looking at here is that when we look at the surface area to volume ratio, it drops off very rapidly as the cell increases in size. And so this is what the graph looks like. There's this exponential decay. And for a cell to be functional, you want to have high surface area to volume ratio, because if you're a cell, there's only so many places where material can enter and exit. And as the cell becomes larger, the ability for those materials to diffuse in and out becomes very limited, and you're not able to have a functioning cell center. So that's why cells have limited cell size. Functions of life, we knew this one as Mrs. Hang, right? So these were the 7 functions of life. They were metabolism, which include things like respiration or photosynthesis, any reaction where you're building or breaking things, reproduction, sensitivity to the environment, homeostasis, where you're putting things in balance, excretion, where you're getting rid of waste, and nutrition, every living thing needs to get food, and every living thing grows. So for the functions of life, this is the mnemonic that we use, Mrs. Hang's: metabolism, reproduction, sensitivity, homeostasis, excretion, nutrition, and growth. In topic 1, we also made some new friends, some protista friends. One of them was the paramecia. And so we had to be able to identify the different parts of the paramecia and what function of life those parts are tied to. So on the outermost, the paramecia has the cilia, which allows it to move. So that is tied to sensitivity to the environment. So these little hairs help them move around. So 2 parts of the paramecia that help control homeostasis, right, keeping things in balance, are primarily the membrane, which controls what exits and enters. And then because these are water-based animals, remember they have the contractile vacuole. So when too much water diffuses in, that vacuole will contract to expel the water. We also had the cytoplasm, which is where a majority of their metabolic reactions are occurring. We have the nucleus, which is tied to reproduction. We have, what else? Our little food granules, which are tied to nutrition. And everything that is a living thing needs to do some kind of excretion. And for the paramecia, they use the anal pore for that. So just to recap there, cilia for movement, membrane and contractile vacuole for homeostasis. The contractile vacuole is homeostasis of water. For the metabolic reactions, we have the cytoplasm for reproduction. We have the nucleus. We also have the anal pore for excretion. And these guys have food vacuoles, which they use for nutrition. So that was everything we had to know about the paramecia. There was another organism that we looked at, the Chlamydia demonis. And so as this, green color implies, this little guy is also photosynthetic. So like the paramecia, it also has a membrane and a contractile vacuole, both of which are tied to homeostasis. And the vacuole is still homeostasis of water. The cytoplasm, where the metabolic reactions are occurring, it also has a nucleus, which is tied to reproduction, and how it responds to the environment. It's also mobile, but this time it doesn't have cilia. It's got flagella, which allows it to interact with its environment. And it also has eye spots to allow it to move towards the light. And so this little guy also has chloroplast, right? That was where that green color was coming from. And so that is where it gets its nutrition from. And so just to recap for this little guy, like the paramecia, membrane for homeostasis, contractile vacuole for homeostasis of water, cytoplasm where the metabolic reactions occur. It also has a nucleus tied to reproduction, and the flagella and eye spots both contribute to sensitivity to the environment. And the chloroplast is how it gets its nutrition. So in topic 1, we also looked at magnification. So this was the Mr. A equation, that magnification equals the size that you measure with your ruler divided by the actual size. And just like before, we always start these problems by going immediately for the scale bar, because that's the only number you have to work with initially, right? So we have this 5 micromolar scale bar here. And let's say I use my ruler and I have my, I don't know, it's like 1 centimeter with my ruler. Pretend that's a cm. How I would fill in this equation is I would put the number I know, my actual size, 5 micrometers. And then I would have my measured size, the one that I have my ruler with, let's say that's 1 centimeter. And remember to calculate the magnification. These are not equivalent, so I have to convert my top number, which is in centimeters, really to my bottom number, which is in micrometers. I take the difference between them, so 2 minus 6 is -4. So for my 1.0 centimeters, 2 minus 6 is equal to 4. Now I have to move this decimal point 1, 2, 3, 4. So now I would have 10,000 micrometers over my 5 micrometers. And this would get me my magnification, whatever number that is, right? So that's how I get magnification. And then typically what these problems are followed by is you'll encounter a cell like this. And again, we always go straight for this scale bar. And I would put that at the bottom, 10 micrometers. And calculate the image size. So that would get me, so I would, I don't know, I put my 1 centimeter, whatever the ruler is, that would get me my magnification. And then what they typically do is they will turn around and say, okay, well, now how big is this nucleus? Right? And so our other equation was the arm. And so that is where actual size is equal to the ruler size over the magnification. So say that I calculated my magnification down here and say it's like 1000x. So arm means I put it on the bottom. I would measure this with my ruler, right, and say it's like 1 centimeter. So if I divide that, that would give me my actual size of that nucleus or anything that I had in between there. So that's how we handle those magnification problems, and you will see them also on paper 3. So for 1.2, that was all about cell ultrastructures. So our primary goal in this one is to identify what cell type, to point to the little organelles and say what they are, and also to draw the prokaryotes versus the eukaryote. So the prokaryote, remember, it has its inner plasma membrane. It's got the outer cell wall, which it uses for protection. It also has that empty space in between, which we call cytoplasm. And because it's a prokaryote, it's going to have the smaller 70S ribosomes, and it will have a nucleoid, not a nucleus, nucleoid, where that single circular chromosome with no protein hangs out. The prokaryotes also have the circular plasmids, which carry some extra genes, and it's got this hair and pili. Now, these are different than the cilia and paramecia. The pili are for attachment, and the pili are unique to the prokaryotes. And it also has the flappy things, right, the flagella, that it uses to get around, just like that Chlamydomonas had as it was swimming around. So just to review, when you're drawing your prokaryote, right? Okay, inner membrane is plasma membrane, outer one is cell wall, empty space is cytoplasm. You draw your pili that are for attachment, these little hair-like projections. The flagella are going to be the flappy tails that help it move around. Draw an area for the nucleoid in the center, which is where its chromosome hangs out. And then the little dots are the 70S ribosomes. And then don't forget your little circles for plasmids. Those are unique to the prokaryotes, right? Okay, so these are the ones, only pili that you're going to see in the prokaryotes. So for the eukaryotes, the first one, the easier one to draw, is the animal cell. So you're still going to have cytosol or cytoplasm. You're still going to have a plasma membrane. That is what stays the same. But now, you kind of like work your way inside out. So the nucleus is the main part. This is where our, multiple linear chromosomes are hanging out. Directly attached to that is the rough endoplasmic reticulum. And then you also have the presence of mitochondria, the site of respiration in the animal cells. And then after the rough ER, a distance away from it, you have the Golgi, which is capturing those vesicles that are sent over from the rough ER for those proteins that are being secreted. And don't forget about your ADS ribosomes. So this is the main difference, or one of the big differences between the prokaryotes and the eukaryotes, is that in the prokaryotes, you're going to have the 70S ribosomes and the ADS ribosomes in the animal cells. So just from the top, animals, no cell wall, only the plasma membrane, cytosol, nucleus attached to the rough ER, which is going to feed vesicles to the Golgi, your mitochondria, which are the site of respiration, and then you have your ADS ribosomes. So plants are pretty identical to the animal cell, except you add a cell wall now. You also have the additional structure of a vacuole that stores water. And of course, you have the chloroplast, which is the site of photosynthesis. And so when you're looking at these, right, I for sure know that immediately this is a prokaryote, or I'm sorry, a eukaryote. So this is a eukaryotic cell, and from where I'm standing, it looks a lot like an animal cell because I don't see any large water vacuoles, and I definitely don't see any chloroplasts. So the large structure in the middle is clearly a nucleus. These little breadbasket things are the mitochondria. You might see them as like these little circular guys. And what is always immediately adjacent to the nucleus are the rough endoplasmic reticulum. And then from there, you might see these big globs, which are probably vesicles that are going out towards the Golgi somewhere. So if you look on this side here, nucleus immediately adjacent, rough ER, mitochondria right here, and then further away, you have those folds, which are the Golgi apparatus. So that's how you know what you're looking at there, right? So this one is clearly a plant cell because we have our nucleus here, which we've seen before, and we have this huge open space, which is, of course, the water vacuole. And then a plant cell is going to have these chloroplasts, which are much larger than the mitochondria. And then what is also different is the mitochondria have these side-to-side structures, right? The cristae, whereas the plant cells have the chloroplasts, and these tend to go up and down. So that's like, those are the two easy giveaways that it's a plant versus an animal cell. Okay, what they all have in common, right? So plant and animals, they're both going to have the nucleus. They're both going to have the ADS ribosomes with the 70S ribosomes in their respective mitochondria and chloroplasts. They're all going to have, because they're prokaryotic, an endoplasmic reticulum, a Golgi apparatus, and the mitochondria. What's unique in animals is we do not have a cell wall, whereas the plant does. The plant is going to have those chloroplasts. It's got those large water vacuoles, and it does have that cell wall. So just a reminder that the big difference between the eukaryotes and the prokaryotes is that those cells are much larger and they have compartments. Okay. So from there we moved on to membrane structure. And so this is the diagram that y'all know how to draw by heart. And so just a reminder that the main component of the membrane is the phospholipid. And it's got two main parts. the head structure, which is hydrophilic, meaning it's water-loving, and then it's got these, lipid tails, which are hydrophobic, right? So they don't want anything to do with water. And so because the phospholipid has both these characteristics, it's considered to be amphipathic. And because of that, it's going to automatically arrange itself into those phospholipid bilayers. So when you mix these phospholipids with water, the hydrophilic head is automatically going to orient itself towards the water, and the hydrophobic tails are going to try to run away. You will automatically form that lipid bilayer. And so another component of the membrane is cholesterol, which controls the membrane fluidity. So it's going to determine how rigid or fluid that membrane is. So the function of membrane proteins, right? So these are things that you have encountered across the course. So you could probably just even talk about specific examples. So some of the membrane proteins that we have encountered are receptors, right? So all those receptors that we have talked about in, topic 6, that we're receiving information from, say, a neurotransmitter, or even those receptors of the hormones, right? So a protein can be a hormone binding site. It can be immobilized in the membrane, like the proteins lipase and peptidase were for digestion. Some proteins can help with cell adhesion. Some of them deal with cell-to-cell communication. So that's what we were seeing in the synapses, right? Receptors receiving those neurotransmitters. That's cell-to-cell communication. So that's a job that a protein could have. We have seen many examples of these channel proteins and the pump protein, right? Especially like the sodium potassium transport. So all of those are jobs that a membrane protein could have. And so for the membrane diagram, remember you had to include one labeled phospholipid. So I label the head and the tail. I demarcate that this is a phospholipid bilayer. I show my integral proteins. I show my peripheral proteins. They're on the edge. They're on the periphery. And I decorate any of those proteins to make a glycoprotein with these like little sugar groups. And it doesn't matter where those are. So we also talked about who came up with these membrane models, right? So the big one is it's Davson and Danieli. So their original model was that it was the protein sandwich, right? Protein, phospholipid protein. And their model was that protein was only on the outside, not in the interior. But this was disproved through the freeze fracture micrographs, where they broke apart the membranes and exposed that there was proteins contained within the membrane. And so that was what led to the falsification of the Davson-Danieli model. So now we have the Singers-Nicholson fluid model, and that's the one that you draw today. So when we look at membrane transport, remember there were two main types: passive, doesn't require energy; active, which does require energy. And so if you think like passive, that is what is going to happen automatically, where you're moving from high concentrations to low concentrations, whereas active, you're moving from low to high. I like to think of this as like the elevator analogy. If you have a bunch of people in an elevator, there's a high number, and as soon as you open the door, they're going to run out the door to a low concentration, versus if I already have 20 people stuffed in an elevator, and in this elevator, I try to add more, you're going to have to really like shove them in there. It requires energy to do so. So our passive transports were simple diffusion. So those are small molecules like water or gases, which go across the membrane with no protein whatsoever. So that is in contrast to facilitated diffusion, where you have larger molecules like glucose or some such, but it's still going from that high to low, only this time you're using a carrier protein to make that movement. So that's what it's different from simple diffusion. Osmosis is the movement of water, right? Isotonic, equal amounts of water in and out. Hypertonic, the water outside is salty or sugary, so the water rushes out. And in hypotonic, the cell itself is sugary or salty, and water is going to rush in, right? And so we saw examples of that a lot of times in the red blood cells, where if it's isotonic, it looks like a regular little donut red blood cell. but when it was hypertonic, they take on this spiky appearance because water is leaving, versus hypotonic, where water is coming into it and causing it to like swell. Our active transports, we're the sodium-potassium pump, right? And so this one we've seen a lot where 3 sodiums are being exchanged for 2 potassiums. We've also seen exocytosis, right? Exocytosis is exiting. You're going outside the cell. And who handles that is the rough ER, where those proteins are synthesized. They package them in vesicles, send them to the Golgi, where they are modified and then repackaged for secretion outside the cell. So exocytosis is for proteins that are exiting. Endocytosis is entering, right? Exocytosis exit, endocytosis enter, right? And so this is where the membrane pinches in and just brings materials in. So these are our 3 forms of active transport. We talked a lot about transport in the axons in our neuron unit, so I'll kind of like save that for that review, well, let's touch on it a little bit, right? Okay. That a sodium-potassium pump is responsible for generating that initial membrane potential. And just a reminder that inside the cell, inside the axon, and that's what we're looking at here, is a negative charge of 70 millivolts. And that is being created because I'm kicking 3 sodiums out and I'm only bringing 2 potassiums in. So which is the greater number, 3 or 2? Well, it's clearly 3. So the outside is more positive, the inside is more negative. That's what's giving you your resting potential. Once the axon fires, right, those charges flip. Now the inside is positive and the outside becomes negative momentarily. And that part of the reset is one of the potassium channels, right? So these guys open up to allow the flow of electrons, and that's part of the reset. But we'll come back to that in Unit 6. So 1.5 is about the origin of life. So cell theory says all cells come from pre-existing cells with really no comment on where that original cell came from. And that originally, right, there was this idea of spontaneous generation, that life can spring up from non-living matter. So we talked about like the recipe. If I want to make a mouse, I can just put out some like wheat in an old rag, and then all of a sudden mice will appear. Or if I want to create maggots, I just leave out, meat, and I can create life from nothing. And that the guy that disproved that was Louis Pasteur. He had his swan neck flasks, and in both of them he has broth, right? Which is basically like bacteria utopia. He boils them both to kill all bacteria inside that apparatus. And that one is broken open to expose it to the air, and the other is left sealed in the swan neck flask. And sure enough, the one that grew it was only the one that was open to the environment. And so this is how, he contributed to the development of cell theory, that only the open flask, the one that was exposed, was able to have like life. So life doesn't spring out of nowhere. It needs to come from a pre-existing cell. So from there we talked about, okay, well, where did the original cell come from? And what we know is that from the Miller and Urey experiments, right? So these were the guys who put like the primordial soup in a flask. So hydrogen gas, methane, ammonia, and shocked it. And they were able to create simple carbon compounds like our amino acids, like ribose. That's what was collected from that. And we also know, from further studies later, that like these carbon compounds, amino acids, and our ribose sugars, and some like bases, show up on rocks from space. So simple carbon compounds can occur in nature. From there needed to be some kind of way to form the polymers, right? So our fatty acids are long chains of carbon. Nucleic acids are long chains of nucleotides. Our proteins are made of long chains of amino acids. So how could you create that from these simple carbon compounds? Well, you could possibly use the heat from the deep sea vents, right? That can help facilitate the creation of those polymers. You also needed to have some formation of membranes, which, as we know, they will automatically form bilayers. So that's cool. And then you also needed to have some mechanism of inheritance. And so there's the theory that RNA was the initial molecule of inheritance over DNA. And so the evidence for this theory, right, that at least we know it's a single ancestor, is that all organisms on Earth use the same 4 bases in DNA, our adenine, guanine, thymine, cytosine, and all organisms also use the same 20 amino acids and the same 64 codons. And so that genetic code is universal. That suggests that there was a single ancestor that developed, however many million years ago. We also talked about endosymbiotic theory, right? So how do you go from a simple prokaryote to the complicated eukaryotic cells that we have today? And so endosymbiotic theory states that you start with, these 2 independent bacteria. So prokaryote 1, who's rather large, and prokaryote 2, who's small and vulnerable. And that prokaryote 1 engulfs that smaller bacteria, but does not fully digest it. And so the reason that this was beneficial was because it ate a prokaryote that did aerobic respiration, right? That was able to make the 36 ATPs with using oxygen. And so this became a mutually beneficial relationship, also known as mutualism, right? And so that was the arrival of the first eukaryotes, where a prokaryote engulfed something that was the ancestor of the mitochondria, right? That did aerobic respiration. And that this occurs a second time, right? Where you have this now eukaryote, which is carrying around mitochondria, and it happens upon a cyanobacteria, which can do photosynthesis. And it, decides to inhale this guy and make friends with it, because now I can make glucose and break it down too. So I have the best of both worlds. So the evidence that this is true is not really necessarily related to the shape, though that was mentioned quite a bit on exams. It's really more that each of these, right? So this is a prokaryote, this is a chloroplast, and this is a mitochondria. It's really that they are all double membraned, right? So chloroplasts and mitochondria have both an inner and outer membrane, which is sort of analogous to the plasma membrane and the cell wall you see in a prokaryote. Like the prokaryotes, the chloroplasts and the mitochondria have their own single circular DNA chromosomes in here. And what's more is that they have 70 S ribosomes, exactly like you see in the prokaryotes. So 70 S ribosomes here, here, and here, all 3. And they all divide through the same method. So even though mitochondria and chloroplasts are inside eukaryotes, they divide separately from the cell cycle. They divide by binary fission, which is exactly how the prokaryotes do it, right? So speaking of cell division, topic 1.6 was all about cell division, right? And so this is our good old friend, the cell cycle. It has a G1, an S phase, and a G2, and this mysterious M phase. So, this is not the one I want, right? Okay, the growth phase, right, which includes G1, S, and G2, is called interphase. Remember that S phase itself is where DNA replication is occurring, and that M is the mitosis, right? That was following the separation of the chromosomes. So cell cycle overall, longest phase is interphase, G1, S, and G2. S phase, replication of DNA. Separately from interphase is M phase, which is mitosis, the separation of the chromosomes. And so this was governed by specific cyclins. And so the word I came up for that was DAB. So cyclin D triggers entry into the G1. And so G1 and G2 are really the same thing. It's protein synthesis. The organelles are increasing in number and size. So cyclin E is going to prepare the cell for entry into S phase. And so remember, DNA replication occurs in S phase. Cyclin A actually activates that DNA replication near the end of S phase. And B, DAP, right, Cyclin B promotes the spindle formation that you're going to need in mitosis to separate those chromosomes from each other. So Diab, cyclin D triggers entrance. Cyclin E prepares the cell for the S phase. Cyclin A actually activates the replication of DNA, and cyclin B promotes that spindle formation that you're going to need in mitosis, right? And so mitosis, just a reminder, gives you two genetically identical nuclei that's different than meiosis, which gives you 4 different haploid gametes. So cell cycle mitosis is for somatic cells. So hair, teeth, eyes, not sperm and eggs. Mitosis is for growth and repair only with genetically identical cells. Okay, and mitosis is also following the separation of the sister chromatids, which you also see in meiosis, and it has those PMAT phase, right? Prophase, metaphase, anaphase, telophase. Okay. So after the cell cycle is complete, to fully separate these two cells from each other, you need to conduct something called cytokinesis. So and it is different in plants and animals. So in animals, it is governed by a contractile ring made of microtubules called a cleavage furrow. And so it contracts and it's going to pinch off the cells from one another. In plants, this is now the Golgi, right, which is sending off little vesicles to create a cell plate in the middle, which will become the division between them eventually. And so that's how plants do cytokinesis. So animals, contractile microtubules to form a ring, a cleavage furrow that pinches off. Plants, Golgi vesicles going to the middle to create the cell plate, eventually creates 2 new cells. Okay, PMAT, mitosis, right? Prophase, metaphase, anaphase, and telophase. In prophase, you might see cells where they're dispersed or starting to condense, but in no particular order. In metaphase, the cells will align along the middle. In anaphase, they will pull apart from each other. And in telophase, you're going to see the formation of 2 different nuclei, right? So that was the whole point of mitosis. You might also run across the mitotic index. So it's a percentage. The number of cells in any stage of mitosis divided by the total number of cells. So if I looked at this graph or this picture and I said, okay, calculate the mitotic index, I would count the total number of cells. So let's say there's 30, just making it up. And so that would be the number on the bottom. Total number of cells is 30. Right, so let's say I have a total of 30 cells. Then for this numerator, I want to calculate how many cells are in some phase of mitosis. So this looks like an anaphase to me, so that would be 1. This one looks like a prophase 2, prophase 3, anaphase 4, some kind of prophase 5, prophase 6, metaphase 7. So I would say, okay, well, it's 7 divided by 30 would give me my percentage, right? So maybe this is, it's whatever percentage that is, right? So that would be my mitotic index. So that's how you calculate those. Okay, so errors in the cell cycle where there is uncontrolled growth will lead to cancer, right? And so we talk about our cyclins, right? Cyclin Diab, right, that go this way. So their job is to promote the cell cycle. So if they have mutations in these genes, it can accidentally, right, drive the cell cycle to occur when it is not supposed to occur. So those are called oncogenes because when they're mutated, they promote cancer. So that's different than a tumor suppressor whose normal job is to stop the cell cycle. Like it detects something's wrong here and it's going to put a brake on that cell cycle. So cancer occurs when you have mutations in both these classes of genes, where you have mutations in your oncogenes, which are going to drive the cell cycle. If there is an absence of these tumor suppressors, then it just keeps going and then the person will have cancer. So where do those mutations come from? So these mutations are happening all the time. They're happening to you right now. and they're caused by these so-called mutagens, right? So these are the biological agents that are going to cause damage to your DNA. And they can include things like radiation from the sun, or if you happen to be standing near, like, I don't know, a nuclear bomb or something, that are going to break the DNA. They can be substances that alter the base sequence. So things like cigarettes, right, or playing with mercury. Those are the carcinogens which are most likely to cause cancer. And if somebody does develop cancer, where it starts initially is called the primary tumor. So this is where the first cancerous cells occur. Once it starts to move, that movement is metastasis, right? So it's going to find other sites around the body to locate, and wherever those sites are replicating, those are the secondary tumors, right? Because they're second. So when cancer starts with a primary tumor. Once it starts to move, that's a metastasis. And once those tumors start to develop at those secondary sites, that gives rise to the secondary tumors, right? And so these are the ones that are potentially like more dangerous and fatal. So that's it for topic 1.